Welcome everyone. I'm David Tóth, sixth-year medical student from the US Lab Biological Research Center. The first question we have to answer: how a researcher works. So we ask questions: what, why, and how we would like to observe. Based on our questions, we plan and perform experiments. And from these experiments, we got results which we have to interpret. With my project, I would like to lead you through this path. Let's start with the mechanism of coronavirus infection. So coronaviruses are invading our cells and steal our endomembrane system to form double membrane vesicles, so-called isolation membranes, to isolate their replication from our immune system. So if you look at this graph or this figure, you might have some questions. Which coronavirus proteins mediate this process? Which host factors can interplay with coronavirus proteins to form double membrane vesicles? A bunch of double membrane vesicles are formed in our cells every day. The phenomenon which processes this is autophagy. Autophagy is responsible for the removal and degradation of useless proteins and organelles. One of the key factors which mediates this process is Atg8, mammalian Atg8 protein, which is required for the early steps of this double membrane vesicle formation, the autophagosome formation. We can distinguish based on the structure 6 different Atg8 proteins. And if you look at this figure, you might see how conserved, evolutionarily, how conserved are these Atg8 proteins. Surprisingly, it was shown that MHV, mouse hepatitis virus, it's a coronavirus family member, replication strongly reduced in those cells which were lacking Atg8 compared to those which harbor Atg8. So this suggests that Atg8 is an essential factor of virus replication. I would like to emphasize that it's not a unique mechanism for coronaviruses, because other viruses are using the same mechanism. If you look at this figure, you can see the SARS-CoV-2 genome, which encodes non-structural proteins and structural proteins. With our bioinformatic analysis, we searched something on these proteins. So what was that? It was the Lear motif. Lear motif corresponds to the shortest amino acid which is responsible for the interaction of a certain protein with Atg8 protein, as the example shows. So my colleague Gábor Horváth used databases to find SARS-CoV-2 proteins which contain Lear motif. And he suggested some, but I would like to point is that there were some proteins in which more than one Lear motif was present. Let's talk a little bit about our experimental setting. So we used MCF-7 breast cancer cells, and we expressed in those cells GFP, green fluorescent protein, tagged SARS-CoV-2 proteins with mCherry tagged Atg8. In the other experiment, we expressed single SARS-CoV-2 GFP tagged proteins with, and we marred the endogenous Atg8. And we were analyzing the colocalization, the interaction of these proteins with fluorescent or confocal microscopy. So if the proteins did not colocalize, what we have seen was single green dots and magenta dots. If they colocalize, what we have seen was white dots. So our analysis showed 71% of real interaction. So we experimentally confirmed our predicted interactions. One example is NSP3, which showed white dots here, if you look at this. And another example for the lack of colocalization was ORF9b, which showed a really interesting phenotype, but there was no colocalization. And I would like to emphasize that NSP3C term showed a really interesting phenotype and colocalized with Atg8. So what was the interesting phenotype? So NSP3C term formed ring-like memory rearrangements independently of the tag, because we used blue fluorescent protein tag, independently of the cell type, we used renal cell cancer cell line, and independently of the species, because we used Drosophila cells here. One of the strongest points of our analysis is that the C-terminal part of NSP3 showed interaction with all MATG8 homologs. Some examples are the GABARAP L1 and the SC3A here. The next question rises how the C-terminal part of NSP3 is able to interact with Atg8. There are two possibilities, as there were two Lear motifs on the C-terminal part of NSP3, and thus we created single mutants and we created a double mutant to answer this question. And what was the result? So the result was that, as I mentioned, there was two Lear motifs, and if we mutated the WXXL Lear motif, the phenotype did not change, so there were ring-like membrane rearrangements. But when we mutated the Lear motif of the NSP3C term, the phenotype strongly changed, so there were no membrane rearrangements, and the ring formation did not happen in the double mutants too. So if you're expert, you can easily interpret these results, but what if you are not an expert? You have to spread your research. How can you do this? You can make a statistical analysis and data visualization, because on this way, everyone can understand and interpret your results. So what can we do with this data? We can calculate the number of membrane rearrangements in the mutants and in the wild type. We can measure the diameter of the membrane rearrangements, and we can make or perform a deeper statistical analysis. What can we do with this 2D picture? If we perform slicing with our microscope, we can make a 3D visualization of these membrane rearrangements, and we can show how they are exactly built up, so how they really look like. And with this method, we can perform colocalization analysis, which is really useful if you would like to show how strong are your interactions. And you can use different coefficients to show this. One of our future plan is to find the exact amino acid which can be responsible for the interaction between the Lear motif. And our collaborators performed sequential analysis of many SARS-CoV-2 isolates, and they found that there are 2 amino acids between the Lear motif which did not change, and there is one more candidate which changed once in that high number. So I think this is a promising line to find the exact amino acid which can be responsible for the NSP3 C term and Atg8 interactions. So to summarize our key findings, 71% of the predicted interactions were experimentally confirmed. The C-terminal part of NSP3 formed ring-like membrane rearrangements independently of the cell type, the tag, and the species. And also, the C-terminal part of NSP3 showed interaction with all MATG8 homologs on Lear-dependent manner. I would like to thank to our lab members, especially to Gábor Horváth, Tamás Marus, Adél Ürmösi, and I would like to thank to our collaborators and of course our supporters. Thank you very much for your attention.